Okay, so good day, everyone. Thank you, Alison, for inviting us to share our experiences and initiatives. Although this is quite specific because I'll be talking about our own initiatives in the university for Eastern Visayas and neighboring regions. Not really the whole Philippines because quite big, but I'll be sharing with you our initiatives from the National Coconut Research Center of the Visayas State University. So I'm a professor there and also an affiliate professor at the National Coconut Research Center. So just a very quick look of our university, just to tell you about University of Agriculture and Allied Fields with 4 campuses. And of course, it's an agricultural land. We also have crops and coconuts there, among others. And basically, I just want to highlight it's a home to the National and Regional Research Centers, one of which is the National Coconut Research Center, which I am affiliated, and we do the initiatives on coconut as our commodity. This is the one, this is the center. It's quite really not a very young center. But this has been established in 1975 as a regional coconut research center and has been upgraded into a national coconut research center, although our focus is only in the Visayas because we do have also other coconut research centers in the Philippines. So you will see that we do have different trusts in the center, but that includes the pest management topic or concern. So why we do have, because of course, just to tell you that of course in Eastern Visayas, it's an agricultural region. And aside from all the impacts of the natural disasters and all other factors reducing productivity, agriculture productivity of the region, I guess one of the factors that also affect the reduction of productivity in the region is that there is a need to address the pressing and persistent problems of coconut pests in Eastern Visayas. Aside from rehabilitating the coconut industry after that very serious Typhoon Haiyan that actually affected the whole region, and also from the aging coconut stand of Eastern Visayas. So the pest problem situation in the region, we do have major pests, as I'm very happy that Mr. Sulab mentioned about rhinoceros beetles, and possibly this is a good way to collaborate with you because this is one of our biggest problems, the persistent rhinoceros beetles we have in the region. And of course, we were affected by the Bruntispa where there was a serious infestation. And now we are trying to monitor and assess this emerging pest, one of which is the Asiatic palm weevil, and of course, this coconut scale insect, or we call it the CSIs. So because of that, we actually developed in the university to answer all these needs in the region, which we would want to help in managing this problem in the coconut industry. We developed a research program with actually broad strokes. Now we want to assess, monitor, and identify these major coconut insect pests in the region and even the emerging ones. And we would like to develop some sustainable strategies using, of course, those successful strategies from other regions, other countries. And there's another one which we have on an ongoing project on the evaluation and development of biocon of Asiatic palm weevil. So we actually tried to focus on 3 major pests now: Bruntispa, coconut rhinoceros beetle, and the APW. So with that, what are the major activities conducted? I don't have to really mention all of this because I'll be sharing with you some of our highlights, or what we have done so far as these 3 different pests impacting the coconut crops in the region is concerned. So we did assessment, monitoring, and then we identify potential natural enemies, conduct biological studies, evaluation in the laboratory and partially in the field or greenhouse, and then develop IEC materials. And among others, we also shared these results to our coconut stakeholders, our farmers, conduct orientation trainings and information awareness drive so that they themselves will help us monitor and even make a detection of what is there in the field. So what have we done so far? So from 2014 to 2022, we actually did regular monitoring and assessment of the damage of Bruntispa in various coconut plantations in the region. So you will see on my screen, I don't have to really go into the details, but we saw that Bruntispa is still present in the region, although the population fluctuated erratically from being high and then also low, and because of guess we know that this is also affected by environmental conditions. Now for rhinoceros beetle, we really see that this is a persistent problem ever since, and the more that we were concerned because after Typhoon Yolanda or Haiyan, we actually did monitoring of some selected areas in Leyte and Samar impacted by the typhoon. And after 2 years, 2 to 3 years, we really saw that almost 100% of those standing trees were actually now invaded or infested by coconut rhinoceros beetles. So just to tell you that there were 33 million coconut trees damaged, and 10 million of these were actually of different severity. So we conducted those areas which were really very highly impacted by the typhoon, and there were a lot of those trees, fallen coconut trees. So this was the data that we got, 100% damage and presence of rhinoceros beetle, and even in the coconut stumps and the fallen coconut logs. So for the Asiatic palm weevil, this is an ongoing project, and we are actually thanking the USTP card for having a funding for us to evaluate biological control or biorational control agents for management of APW. And we saw that there is an increasing. So I guess this is our next problem now would be the increasing number of infestation of APW, Asiatic palm weevil, in the region. So we have already reported incidents of increasing incidents of APW in Leyte and Samar provinces. So we really hope that after the project or through the project, we would be able to connect or collaborate with other agencies and could help us whatever the results and outputs of our research project. So as far as the CSI or the mealybugs of coconut, we actually just, we had one in, incidents of infestation, and that was somewhere in southern Leyte. And through the help of or partnership with PCA, we were able to at least do some management strategies in order to reduce the infestation or even manage the infestation of CSI in the southern Leyte portion. So now there has been no report of increasing infestation of CSI in Leyte. So for just to really understand all these major pests and natural enemies, we did some biological studies. So we did biological studies of Bruntispa on recommended varieties in the region, like our tall and dwarf varieties, just to understand what's the development of this Bruntispa in this different recommended varieties, so that we will be able to really know how to manage them as far as these different varieties are concerned. We also were able to study a little bit on the alternate host of Bruntispa, which we could incorporate in our management strategies in the future for Bruntispa. And we saw that among the palms, coconut-related plants or the palms, there were some of those which actually we're able to support the Bruntispa up to the adult stage. And that could actually be used to also strategize our control measures. So for the natural enemies, we actually were able to discover some of these natural enemies, including these parasitoids, predators, the earwigs, and some EPN, although we haven't really completely fully studied the all the biological information of this. And we also did some studies on Beauvaria, Metarhizium, and Licanicilium. These were some potential EPF. And we found, later I will have a slide to show what were the R&D results that we have. So for rhinoceros beetle, we were actually able to find some of these, like the EPF. I cannot, okay, here. So, so sorry. So these were the potential natural enemies that we found collected in the field, including a nematode, and not really much on the virus, but we also found some entomopathogenic fungi, and these were the Metarhizium anisupleae, which we used in screening, or we did some laboratory screening. So for the laboratory evaluation of these potential biocon agents, we did also understand the biology of Tetrasticus by knowing at what various ages of the pupa of Brontispa is really the effective host. And then we also did the field parasitism of this Tetrasticus, because we also did some releases of these parasitoids in order to really help the infestation of Brontispa in the field. And we did the laboratory evaluation of fungi against, or EPF, Brontispi adults and larvae in the laboratory, and we found that there is really an efficiency, although of course we really need to really confirm and validate our data more, but we found that this EPF were actually effective against Brontispa larvae and adults. So for, okay, I mentioned that we did releases of our parasitoids in the field. So we actually, because there were a lot of problems, or there were requests on how to manage their Brontispa infestation in farmers' field. So what we did is we did a mass production of biocon agent, and particularly the Tetrasticus. We have sustained mass production of these in our laboratory in the Coconut Center, and also for the mass production of airwig to really augment the control in Brontispa in the field. So until this period, we have actually distributed 40,000 mummies. The mummies of the pupae, which were actually parasitized by Tetrasticus, we have produced this, and then we actually distributed it to coconut farmers in various parts in eastern Visayas and other parts of the Visayas Islands for releases in their farms. Although our limitation is that we could not produce as much that we would like, because we would the limitation of the pupae of Brontispa, and we probably would explore, or the I guess our alternatively would like to explore alternate hosts for the mass production of parasitoids, and this could be our future studies. So with that, in order to really monitor how efficient and effective our parasitoids, we also did recapture and recovery studies of our parasitoids in the field, and we did some of this actually testing in the field, no, their efficiency. So we released parasitoids in the field, and we saw that there was really an their effectivity was shown, because some of these infested palms, the makapuno palms, has actually recovered from the from Brontispa infestation. Also, for rhinoceros beetle, we also did studies on prevalence and occurrence of entomopathogens infecting coconut rhinoceros beetle, and we did have encountered some larvae of coconut rhinoceros beetles infected by the EPF, the pathogen, the Metarhizium. And actually, these were also, we set up lag traps in order to also trap those adults and see the infection, the EPF infection of the larvae that are actually present in the lag traps. Although our ongoing initiatives now is also to mass produce and evaluate these pathogens, entomopathogens for CRB, and we have encountered problems on high contamination of cultures, and our current and ongoing project, which we are actually also conducting now, is to identify, isolate, which is more really very effective for our own population. And as mentioned by Dr. Sulaf earlier, we really need to characterize our CRB population and their associated entomopathogenic organisms, because we have instances wherein this EPF actually were not, there were no infections in our bioassay for the EPF. So future study activities that we want to conduct, we would like to, I mentioned this already, identify and maybe characterize our CRB population and the associated EPF, and integrate the use of lures or pheromone traps for adults, which we have tried, but we really need to do more of this. Although we have challenges in securing or accessing our pheromone traps, because of, I guess, resource constraints. So another one would be to explore indigenous control tactics utilized by coconut farmers. Although we have asked them, but they do have, but we may have to really explore more if they have indigenous control tactics, and maybe other biorationals or natural products which we could use as potential control of our CRB or managing CRB. And another future activities that we would like to conduct would be start documenting coconut diseases, although these are already big projects that we have, but there has been no initiatives so far on coconut diseases affecting the coconuts in the region. So
10 seconds, Juliet.
yeah, other activities that we conducted actually is we develop IEC materials on site-specific sustainable management of our coconut insect pest. This is very helpful because we did orientation trainings and information awareness drive of coconut on coconut insect pest to our farmers and our coconut stakeholders, and even the those staff of the Philippine Coconut Authority of the region. We also did site visits to assess and provide technical services to coconut farmers. We do get some requests and questions or queries from our coconut farmers, especially related to their coconut pest problem. And that is also what we did. So this is just to show that right after Yolanda, we did a lot of intensifying our information drive, especially with the severity of the infestation of CRB, and at the same time the CSI during that time, and also the Brintispa infestation. So there were a lot of information drive that we had in the region, although really concentrated only in Eastern Visayas. So I guess with that, thank you very much. These are the initiatives that we have, and I thank Allison for inviting us again, and maybe make partnership and collaboration with the previous speakers. I see already some potential collaborations which we could request from our previous speakers. Thank you very much.
Thank you so much, Juliet. And yes, definitely. Listening to your presentation, I was just thinking, wow, you're doing so much work, and so much work that is of interest to the Pacific region speakers that we've just heard from. And I'm sure also that's so true for other speakers that I couldn't invite because we just didn't have enough room from across Southeast Asia as well. So I was sort of struck, this is exactly what this sort of session is meant to do, is sort of start highlighting some of those areas. You're doing a huge amount of work. I've got a question here. So you're doing so much work. Are you working with researchers in other countries as well? Like what we've identified here is there's similar interests working on similar pests. Are you doing that?
Yeah, actually, our ongoing project on CRB, one of our partners is from Indonesia, which we actually are ongoing now. Our project on internationalization. So it's a specific project, and although I'm not particularly involved in that project, but I know that we have initiatives, and this is our internationalization project together with Indonesian partners, and we wanted to work on the CRB. coconut
Ah,
rhinoceros
that's really
beetles.
interesting. Okay, that's great. So it's nice to connect, obviously, your learnings and from that project, hopefully, into the CIB work in the Pacific
Yeah.
Island countries. That's excellent. Another question here. You mentioned a lot of materials that I thought were really interesting, site-specific IEC materials, which I find quite fascinating. Are they publicly available? And then are we able to, are other people able to sort of see how you've developed those and what you've developed?
Yeah, we actually, after the Typhoon Haiyan, we wanted to develop a very, especially on rhinoceros beetle, how the farmers could actually manage their coconut rhinoceros beetle problem. So that is one. And we also had for Bruntispa,
Yeah.
pest. So we also had that, how they could also, like the management of Bruntispa, specifically in our area, where we have all these releases of parasitoids and also potential use of predators, when to release and how to release it. So we have specific material on the, and even how we could teach them how to mass produce, because very easy, they can mass produce the parasitoids. So we had some IEC materials on that, including even the prevention of, or how we could reduce and manage, like the, or how to recognize, especially the coconut scale insect, which was a problem in the Philippines one time. So with that, there were actually some materials that we also did develop.